Well, hello, listening audience. I'm Tom Romito, and we're not quite ready to begin, but I just want to welcome you. So far, I see Chris Johnson on the meeting. Welcome, Chris. Chris is one of our board members at Friends of Ottawa. Marianne Nolan, welcome to you, one of my Cleveland friends.
Hi, Tom.
Let's see who else is here. Ruth Scully. Ruth, I know you, but I don't remember what your connection is. Welcome. Paula Lozano, welcome again. Paula is one of my Cleveland buds in the birding network. Okay, we're up to 15 and climbing.
Distracted by my busy bird feeders. Got a lot of happy jays today.
Yeah, good. Okay, gang, it's the top of the hour. Let's see. Oh. Okay. Rebecca, would you keep your eye on the waiting room for me?
Absolutely.
Swell, thanks so much. Okay, gang, welcome everyone. Welcome to the 2021 Virtual Spring Nature Festival, sponsored by the Friends of Ottawa National Wildlife Refuge. I'm Tom Romito and your host for this program. I'm a board member of the Friends of Ottawa National Wildlife Refuge, and we are the fundraising arm of the refuge. We're a 501c3 nonprofit organization established in 1997 to support Ohio's only national wildlife refuge complex with youth development, public use projects, and most recently, land acquisition and restoration. The refuge is located in the western basin of Lake Erie, halfway between Port Clinton and Toledo, in some of the most critical wetland habitats in the world. If you're interested in learning more about us and what we do, please look at the link that I have already placed in the chat box. In the chat box, you're going to see 3 links right now. friendsofottawanwr.org is our website. The next one, friendsofottawa.org Seek Refuge Days, is the schedule for the rest of this festival. And the third one is the survey we're going to ask you to take at the end of the presentation to kind of rate us on how we did, what you thought of us. The website, by the way, will point you in the right direction to become a member, a volunteer, make a tax-deductible donation to support our work, and shop on our online nature store. Our store.
Tom?
Yes, ma'am.
I don't see anything in the chat box.
Okay, let's see what's going on. I sent it to everyone. Rebecca, could you check that?
I don't see it either.
All right.
It went astray.
We'll do it again. No problem. We're going to do it again. Let's try it again. Does anyone see it now?
There it
Yes.
is.
Good, thank you. All right. So anyway, today we're going to take a deep dive into an interesting aspect of ornithology with Courtney Brennan. Courtney works at the Cleveland Museum of Natural History, where she's currently doing research in Passerine hybridization and migration studies in the Great Lakes region. She's also involved in Lights Out Cleveland, studying bird strikes on windows in the city. Courtney's a Cleveland native and holds a master's degree in environmental science from Cleveland State University. So before we begin, I ask that you stay muted to minimize background noise for our presenter. Please type questions in the chat box as we go, and we'll have time for Q&A during and at the end of the program. So I'm now going to turn the program over to Courtney. I'm going to mute myself. Courtney, you're on.
All right, thank you so much, Tom, and thank you everybody for joining me this afternoon on this thankfully not snowy and sunny day. Hopefully Earth Day will bless us with some snow melt. Okay, so everyone loves a good outline slide. So I'm going to give you a quick background about myself and my role at the Cleveland Museum of Natural History. And then we'll dive into the current research that I've been working on with hybrid birds. And so the bird that started it all. So as Tom said, I was born and raised in the Cleveland area, and I've always described myself as a naturey and an outdoorsy and a sciencey kind of person. But I wasn't quite sure what I was gonna do with myself after high school. I enrolled at Cleveland State University in 2006 with the idea of becoming a nurse. And I truly believe that being a nurse is a calling, and I don't think I ever got that call. And my life kind of completely changed on a family vacation on Kelly's Island, where my cousin took me bird watching. And for the first time, I really got introduced to the world of birds. And I probably don't have to explain what a spark bird is to this group of people. Of course, everyone in this chat probably knows. The spark bird is that one bird that kind of started it all. And for me, that was a cedar waxwing. And after I saw that bird on Kelly's Island, I was hooked. And so everything kind of started changing for me once I became obsessed with birds. So I joined the closest Audubon Society chapter, which was Black River Audubon Society. And I was introduced to landlording purple martin gourds, which was a really life-changing experience to me. I thought that was so cool. During this time, I also volunteered at the Wildlife Rehabilitation Back to the Wild. And that's when I started realizing I really wanted to do something with birds and not necessarily become a nurse. And so I made it official and I changed my major at CSU to environmental science. And then I took a summer job as a field tech where for the first time I was feeling kind of professional identifying birds. And I also learned how to do some plant identification and surveying. And I learned how to become a vacant lot rapid assessment protocol personnel. And I kind of started gearing my way towards going into a research type of career. And then my last semester as an undergrad, I did an internship at the Cleveland Museum of Natural History. And that kind of, set the stage for the rest of my career as of now. And that was the first time I was introduced to the world of specimen preparation. After my undergraduate degree wrapped up, I gathered the courage, to ask Dr. Andy Jones at the Natural History Museum to be my advisor in graduate school. And together we worked on the project of singing behavior and geographic variation in the songs of the Veery. And so after graduate school wrapped up, I had the opportunity to be an employee at the Natural History Museum, which was my master plan all along. So I started out just as a specimen preparator for the ornithology department, where I was prepping birds all the time. And I look back at those years as the glory years. I was promoted to collections manager of ornithology, and then recently promoted again to senior collections manager of zoology. And so as specimen preparator, my main roles were, as you could guess, was preparing specimens, but I also started coordinating the weekly volunteer sessions. And I was starting to learn some curatorial tasks, and I do field work, and outreach is my favorite part of my job. And so you can see this picture of me preparing a bird specimen for a bunch of children on the scale of like really interested to completely grossed out. Here's kind of a dated picture of the volunteer crew, myself, and we even turned it into a family affair, and that's my sister, who's also a volunteer at the museum. As collections manager, my role as preparator expanded with more emphasis on doing a complete digitization overhaul of our research collections. And it was when I became collections manager, I started getting involved with DNA research and becoming a DNA lab tech. on top of the other stuff that I was already doing. And then as senior collections managers, my role has just expanded. So I've been living in the bird bubble up until now, and now the skills that I've developed as the ornithology collection manager, I use to oversee other departments at the museum, including the vertebrate zoology research collection and the invertebrate zoology research collection. But fear not, I'm still an ornithologist at heart, and all of the research that I do is all bird focused, because no matter what, you can't tear me away from those birds anymore. And so now to get into the research part. And so in the DNA lab, the work that we focus on is mostly on determining hybrid parentage of birds that we suspect to be hybrids, and studying genetic variation across breeding ranges of several different species of birds. And so the kind of stuff that I do in that lab include DNA extraction, so getting DNA out of feathers, blood, tissue samples, and sometimes even toe samples. So in our museum collection, we have some bird specimens that are over 100 years old, and we don't have a modern tissue sample for those birds. But even with just a tiny little bit of that toe pad from the study skin, we're able to get DNA out of it. So it's pretty powerful. I also perform PCRs, or polymerase chain reactions, which is a very fancy way of saying I amplify very specific parts of a bird's genome. And then we're lucky enough to have Case Western Reserve University right down the street from us, and they actually have a sequencing lab. So when we get our PCR products, we just take it over to Case, and they can sequence all of our samples for us. So lots of fun action right here in Cleveland. And so the current research that we've been working on has been about a bunch of hybrid warblers. We've kind of turned into the hybrid warbler lab the last couple of years, and it's been a lot of fun. So on this slide, I'm just kind of showing you a list of the different projects that we either have worked on or are in the process of working on. And I'm showing you 3 pictures of the birds that, some examples of some of those hybrid birds. So up here, we've got the northern Parula cerulean warbler. We've got our field sparrow, clay-colored sparrow hybrid down here in the middle, and then a western kingbird, Couch's kingbird over here on the left. And so lots and lots of hybrids. It's been a lot of fun. And so now we're going to get to the latest hybrid story we've been working on, and it's the story of 2 different birds. And so a few years ago at Black Swamp Bird Observatory, during normal spring migration bird banding operations, the skilled and talented bird banders over there caught a very strange looking warbler. That's the one down here on the left. And we've worked in the past with Black Swamp Bird Observatory of different projects, and they know us and we know them. And so when they caught this strange bird, they immediately connected with Andy and myself. And unfortunately, we didn't, immediately start working on this bird. Lots of projects kind of, hit the back burner sometimes just because, work kind of keeps trudging along. But our interest in this hybrid got revitalized when some local bird watchers in a community of Laval, Québec, Canada, discovered this strange looking hybrid, and they connected with the McGill Bird Observatory bird banders and researchers at McGill University. And so this bird was found by bird watchers, tipped them off to people that hold the permits to actually catch the bird. And so we both kind of ended up with research material from bird banding stations the same way in Ohio as they did in Quebec, which was pretty cool. And so based on plumage features alone, we both thought that we had a hybrid bird. And we had a big phone conference with the McGill University and Bird Observatory folks, with myself and Andy Jones at the museum and the folks from Black Swamp Bird Observatory. And we decided, instead of competing to publish these findings, we decided to collaborate together to bring this new hybrid pairing to light, which at the time, no one has ever described this cross before. So it was pretty exciting. And luckily, since it was kind of an incidental kind of encounter, most of the field work was already finished. So when these birds were caught in the mist net in both Ohio and in Quebec, body measurements were taken, they were photographed extensively, and each bird was banded with a little unique number on a little aluminum bracelet. And then feather samples were taken from each bird, and then the bird was released. So fingers crossed that both of these birds are having a safe migration right now. And the wonderful thing about having this collaborative effort with bird banders is they were able to very eloquently describe the plumage analysis portion of this manuscript. And so with these pictures you see on the right, so in this first column here, A through D, those are the Quebec hybrid photos, and E through H here, those are the Ohio hybrid photographs. And so basically we just kind of pitch why we think this is a cross of a Magnolia Warbler and an American Redstart, including the Magnolia Warbler plumage features like the white eye arc, the yellow wash on the belly, the narrow wing bars you can see, and the black lures. And then for the Redstart, these photographs here are kind of difficult to see, but they've got that orange tinge kind of in their armpit area. They've got yellow at the base of most of the secondaries here. And of course, these big patches on the outer tail feathers are all pretty big flags for we've got a Redstart in here somewhere. And so back in the day, this kind of plumage analysis would have been enough evidence to present this and publish it. But now, since there's such advancements in the genetic side of doing research, that we could really make a very compelling story if we back up these plumage characteristics and our plumage analysis with actual DNA lab work. And so that's where my big role came in. And so we developed the protocols in the lab at Cleveland Museum of Natural History and the lab at McGill University. We did the same lab protocols for extracting the DNA out of the feathers of our punitive hybrids. And then we both labs performed the PCR analyses. And for this project, we had to amplify a mitochondrial gene, which we did the ND2 gene, to determine the maternal ancestry. And so mitochondrial genes are only inherited from your mom. So if we can match up a mitochondrial gene between the Redstarts and the Magnolias, we'll see who it matches. And then to figure out who dad was, it's a little more complicated. We have to amplify nuclear genes. And so we amplified the myoglobin and the musk gene for this. And nuclear genes, you get one copy from mom and one copy from dad. And so it's a little bit messier, it's not as straightforward, but that's the way we determine paternal ancestry. And we do that by comparing the genes that we amplify from our hybrid birds to a bunch of other Magnolia Warblers and American Redstarts. And so what I mean by that, we compare them to a bunch of, Redstarts and Maggies. And so here's what our genetic analysis looked like. So we've got the sequences from our Ohio bird and from our Quebec bird. And then we compare those sequences to several individual American Redstarts and several individual Magnolia Warblers from different geographic locations. And where we wanted to include birds from different geographic locations so we could kind of account for the natural genetic variation that happens. So every single human isn't exactly the same for birds. So we wanted to get a little bit of that genetic diversity that may or may not be present in these populations. And so that's the great thing about working in a natural history museum and having friends that also have natural history museums, because we are able to glean these samples from our own collection. So CMNH stands for Cleveland Museum of Natural History, and where we wanted to fill in those gaps. So we really wanted to have some Redstarts from Alberta, and we didn't have those in our collection, but luckily our friends over at UMMZ, the University of Michigan Museum of Zoology, had those samples, and so we borrowed them. So the collaborative effort expanded even a bit more for this project. And so we'll do the easier one first. So the maternal ancestry. And so spoiler, the Magnolia Warbler was the mom. And the way we figured that out was we amplified the ND2 gene, which was inherited by mom, and that is a 1041 base pair gene. So it's a nice long strip of information. And what we do is we amplify that gene for our hybrids and all of those individual Magnolia Warblers and those individual American Redstarts, and we look for variation in that gene. And out of over 1,000 base pairs, there were 80 sites where there was variation. And so we've got special programs that can actually highlight where there are differences in these sites along the gene. And out of those 88 sites that were variable, 77 of those sites were identical among the hybrids and the Magnolia Warbler, and they differed from the American Redstart sequences. And so that's what this graph is depicting here. And so these numbers across the top, that's the base pair numbering. And so as you can see for base at this variable site on base pair 45, all of the Magnolia Warblers and our Ohio hybrid and the Quebec hybrid have an adenine or an A in that position for 45, where the American Redstarts all have a guanine or a G. And you can see across all of these orange highlights, you'll see the Magnolia Warblers and our hybrids are identical, and it's different from the Redstart. All the G's for Maggies in our hybrids and C's for the Redstarts. And so what that is showing us is that the Magnolia Warbler was the mom for both of these hybrids, and that's where they were inheriting these mitochondrial genes from. So that was very exciting for us to find out. And so the paternal ancestry, spoiler, the dad was the father for both of our hybrids. And I told you before that we amplified 2 nuclear genes for this analysis, the myoglobin gene and the musk gene. Unfortunately, the musk gene turned out to be completely useless for this project. It didn't tell us anything informative, but luckily the myoglobin gene was a little more helpful. And so myoglobin is an 801 base pair gene. And just like with the hunt for the maternal ancestry, we look for where there's variation across these genes. And so what we look for the nuclear genes is what's called fixed differences between parental species. And out of 800 base pairs, we found 5 fixed differences. But luckily, they were very informative. And statistically, we had enough evidence to kind of state that we were confident that American Redstarts were the father of these hybrids. And so much like the graph I showed you before, across the top here are the base pair sites. And so you'll see here on 36, we've got our hybrids in the middle, Redstarts on top, Magnolias are on the bottom. And you'll see the Redstarts at site 36 have double A. Magnolia Warblers at site 36 have a double C. And you'll see our hybrid has got 1 copy of the A and 1 copy of the C, presumably 1 from the Redstart, 1 from the Magnolia. And we see a similar pattern like this on all of these highlighted fixed difference sites. So double T's for the Redstarts at site 121, double G's for the Magnolias, and our hybrids got 1 copy of each, 1 G from the Magnolia Warblers and 1 T from the Redstarts. And so with that evidence there, we could say genetically we can back up this hybrid parentage. And so here's just kind of a visualization of what I just said. So we've got the evidence with just looking at the birds, all of the signs that these are a hybrid of a Redstart and a Magnolia kind of come through with just the plumage of these birds. But now we have got the genetic analysis to back it up even further, which makes this a very compelling case. Well, one thing kept nagging at me while I was writing up this manuscript was, why do our birds look so different? And so I failed to mention this before, but the bird watchers rather, that noticed the Quebec hybrid, they noticed this bird because it was singing the song of kind of a strange American Redstart, and it was playing the role of a social partner for a American Redstart female that was tending to a nest. And so just based on that behavior alone, it was pretty, obvious that this was a male bird. But the Ohio hybrid was caught on migration, just in a mist net, just doing its thing. And based on the preliminary looks of the bird, we said, it's probably a male, but we weren't quite sure. And I just had a hard time, writing up this manuscript without really, dealing with why they look so different head on. So luckily we've got genes to the rescue. And so I just amplified their sex gene to genetically determine the sex of the Ohio hybrid. And so unlike humans, the male birds are the homozygotic individual with their sex chromosomes, and the females are the heterogynamic, or homozygotic for the males, sorry. And so if you amplify their sex chromosome and you illuminate the PCR products under a special kind of light, 2 bands will show up for a female and 1 band will show up for a male. And so it's a really simple lab technique. And so I just took a little bit of that extra template that we had when we did the initial extraction for these birds. And what do you know? Our birds look so different because the Ohio hybrid was a female and the Québec hybrid was a male. And so I thought that was such a cool little extra bonus layer to this project, was not only do both of these hybrids have the same combination of a dad American Redstart and a mom Magnolia Warbler, but we get to see the plumage of both a male and a female hybrid, which I thought that was just so cool. So that leads to the question, why on earth did a Magnolia Warbler mate with an American Redstart? The definitive plumage of these warblers are so different that it seems like plumage alone would be a pretty good barrier to keep these 2 species from hybridizing. But as many of you experienced birders might know, it takes some time for birds to kind of grow into that definitive plumage. And a lot of field guides sometimes only put the definitive plumage of an adult male in some field guides. And so what's pictured here on the screen, we've got a young male redstart, which you could kind of argue sort of maybe looks like a Magnolia Warbler. Maybe if you kind of blur your eyesight a tiny bit. And then, I mean, at least to me, and I'm sure some of you would agree, the songs of a Magnolia Warbler and American Redstart can be very similar sometimes. And there's actually studies that suggest that there is like statistical reasoning that can back up that statement, that they do have similar song structures, similar frequencies, similar accents in parts of their songs. And so you could make the argument that their songs are similar enough that maybe there was this mismatching in breeding. Their breeding ranges overlap significantly. Redstarts and Magnolia Warblers populations are stable, if not growing, and they overlap a lot in their breeding ranges. So it's not like these birds aren't in contact. And something else to consider, our male redstarts tend to be kind of sneaky snakes, and they've got polyamorous breeding behavior. And it seems to be a little more prevalent in redstarts when compared to other warbler species. And so that's something else to consider, that maybe we had an immature male, maybe couldn't get the prize females of the bunch, and so kind of took what he can get. And then the other factor is warblers hybridize at a higher rate than any other bird family. And over 50% of warbler species have been documented in some kind of hybrid combination. And I honestly think that 50% is now an outdated number. I think it might be closer to 60 or 70%. Is that a matter of, are we just noticing them more because these are very brightly colored, brightly songed, fun birds that people get excited about? That could be a reason. And so why so many hybrids lately? And because it seems that the sightings in the literature describing new hybrid crosses has been increasing. And what are the reasons for that? And is that actually happening? Well, perhaps, but I think these other reasons might shed some light on that. There's an increased interest in bird watching, and the technology associated with bird watching gets better and better. And so back when I first got into bird watching, it was carrying around a field guide and scribbling some notes on a piece of paper. And now that has evolved to, I've got a field guide on my smartphone, and I can take a quick picture with my smartphone through my binoculars and get a quick picture of a cool bird. And with community science apps and social media especially, this word travels really fast. And so like I said before, the Quebec hybrid was first discovered by 2 local bird watchers, and they connected with researchers. Platforms like Twitter, it connects a lot of birders together, and Facebook groups where people can discuss birds are very popular and have replaced listservs more or less. And news travels very, very fast. And so we actually connected with the folks who found a presumed white-throated sparrow song sparrow hybrid just from seeing it on Twitter, and we reached out to them and connected with them. And as many of you know, news about a strange bird travels very fast. But kind of a more sad note is the sad fact that breeding ranges are changing, and climate change is having impacts as well. And so breeding ranges for many birds are shifting, expanding, contracting across different, new areas, and the habitats change, and they take those birds with them. And then the fact of the matter is some species are coming into contact in ways today that they haven't historically, and that could be another reason why we might be seeing more hybrid events. And so with that, I would be happy to take any questions. I know I just kind of splurted out a ton of information, but I want to give a quick thank you to my collaborators at the McGill Bird Observatory, McGill University, and the Black Swamp Bird Observatory. And with that, if anyone has any questions, feel free to ask.
Ladies and gentlemen, I don't see any questions on the chat room. So this would be an outstanding time for anybody who's got a question to simply unmute yourself, and you can do that either by toggling the switch in the lower right of your screen or by pressing your space bar on your computer and ask your question. Go right ahead. We'll wait and see who's inspired to do that.
All right, we already got one question. So Fred asked, will these hybrids be fertile or sterile? So that's a great question, and we don't know the answer to that. We wouldn't really be able to know the answer to that unless they were studied specifically during the breeding season. And so I mentioned that the Quebec hybrid was seen tending to the nest of a breeding female, but for all we know, all the eggs or young in that nest could have been from a different male, and our hybrid might have just been acting as a social partner. So it is possible, but we really don't know unless that was the focus of the research project, because there's specific paternal analyses you have to do to actually figure out if that is the dad of those offspring. So more research to be done. What am I working on now? Right now I'm working on getting back to the museum 5 days a week. So we're still working in pods due to the pandemic. So I'm only on site on Tuesdays and Wednesdays. And so when I'm in the museum, I'm very busy, but one of the projects we just wrapped up was actually helping another researcher who's over at the University of Toledo, who studies which species of it? Gravireas and red-headed woodpeckers. And so we were asked to determine the sex of those birds that they're researching, because you can't tell if it's a male or female just by looking at it. So I'm anxious to get back to some more hybrid warbler or hybrid bird work, but we might have to be a little more patient until I can get back into the museum full time.
Well, Courtney, you can see another one. If a hybrid does breed, how complicated would it be to establish parentage?
So that's a great question. So I don't know if any of you folks heard about the really interesting hybrid that was a triple cross. So it was a blue-winged, golden-winged warbler crossed with a chestnut-sided warbler. And so it was like a three-way cross, very complicated. And it gets harder and harder to determine the parentage. And so the work that we do focuses on looking at little chunks of a bird's genome. But now there are techniques where you can look at an individual bird's entire genome. And so that's the way researchers are untangling these more complicated hybrid parentages by looking at their entire genome and comparing it with punitive parent species. So it's out of the reach of what I can do in our lab right now. But luckily there are a bunch of researchers that are doing that kind of work today.
And Courtney, Paula says it's interesting about the red-headed woodpeckers.
Yeah, I love doing a fun little collaborative projects for folks, and I'm excited about that project as well.
And Mary asks, Stacy says, I'm about 20 miles from the Western Foundation for Vertebrate Zoology. Do you collaborate that far afield?
So sometimes we collaborate with folks that have samples that we don't. So a big part of my job is I facilitate tissue requests and specimen requests from other researchers. So much like we had to ask the University of Michigan Museum of Zoology to borrow specimens, folks do that with our collection as well. And so there are websites like VertNet and GBIF, where research collections and institutions that have research collections can actually have their information published. So a lot of the times researchers find out about what we have in our collections just based on searching what's available. And so I might not even know the folks that I'm reaching out to, or I don't know the folks that are reaching out to me, but that's how a lot of those connections are made today. We all want to share our research collections. We don't want to hide them away in the basement of museums forever. We want this kind of data to be available. And so, yeah, sometimes I'll be sending tissue samples all over the world.
Well, Courtney, don't see any more questions right now about hybridization. So I'm going to ask you to switch gears and talk a little bit about bird strike science. I know that you are actively involved in Lights Out Cleveland, and you have a team of, there is a team of volunteers that do this. Tim Jasinski from the Lake Erie Nature and Science Center is kind of the coordinator of that. And you take into the museum the live casualties and treat them and maybe release them, and you use the dead ones for research. Can you just talk any more about that, please?
Yeah, absolutely. And so if anyone's unfamiliar, the Lights Out Cleveland program is working towards making Cleveland a safer place for migrating birds. And so many passerine birds, they migrate at night and they use the stars to navigate. And if it's cloudy or there's any kind of overcast situation where it makes it difficult to see the stars, at night, our cityscapes become extremely disorienting and actually draws birds towards the lights. And so, unfortunately, this results in thousands of birds colliding with the skyscrapers downtown. And so there are teams of dedicated volunteers that walk routes downtown during peak migration in spring and fall, and they're picking up injured and dead birds. And like you said, the birds that are injured, they go to Lake Erie Nature and Science Center, where they kind of get a little spa treatment, and hopefully they get released and sent back on their way to continue their migration. And if they die or if they're found dead, they get brought to the Natural History Museum, where they get processed into research specimens. And so we've got two groups of volunteers that work on this effort. Unfortunately, the number of bird casualties received will always be playing catch up, but we've got a group of two groups of volunteers that skin birds and skeletonize birds for our research collection. And so what we're trying to do is build up enough data and build up a case to present to the building managers and owners to get them to, one, turn off their lights at night during peak migration, because if the lights aren't disorienting the birds, the problem gets kind of dealt with, and the birds might not even have to like pass through downtown Cleveland. They can just carry on their way and not skip a beat. And because of the amazing data that these volunteers are collecting, we're learning a lot more about what's actually happening with these birds. And so at the beginning of this, we thought, well, the first route that's walked first thing in the morning, they'll find all of these dead and injured birds, but that wasn't the case. The volunteers were finding out that although, yes, there were casualties found first thing in the morning, some of these birds they were finding them dead after daybreak. And so unfortunately, what happens is the lights draw the birds in at night. Some of them either fall from exhaustion just from flying around in disorientation, or they come down into the little green spaces in downtown. But once that sun comes up, those buildings become as reflective as mirrors, and the birds are colliding with reflections of green space. And we know that because our volunteers are writing down where they found the bird, what building they collided with, what side of the building they collided with, and what time the bird was picked up. And so we started seeing these really interesting patterns of birds colliding into these windows at 8 a.m., 9 a.m., 10 a.m. And so the real danger are these reflective glass structures throughout downtown. And because we know what side of the building these birds are being found at, we can identify the more dangerous facades of some of these buildings. So for example, all of the skyscrapers that face public square, face that green space, have higher bird casualties, because the birds will be hanging out in that green space and they'll say, okay, I'm gonna go fly to that next patch of green, not realizing that they're flying into the reflection of the green space that they're already in. And so although this is kind of a sad program to be a part of, it kind of seems like a never-ending effort to make it a safer city. We are making progress. Lots of buildings are signed up, and we're learning a lot about these birds, and it is benefiting our research collections as well.
Yeah, Courtney, thank you for that explanation. I'd like to return to the issue of hybridization because we have a question for you. I've done most of my birding along the intergrade zone of black-capped and Carolina chickadees. How much hybridization occurs there? Will they ever be lumped into one species?
That's a really good question. So there are many examples of established hybrid zones and hybrid birds, like black-capped and Carolina chickadee, where it's very difficult to know which bird you're looking at in certain spots. And in these regions, it's a well-documented phenomenon that certain species will hybridize with each other. The question of will they ever be lumped into one species? So most scientists can't even agree on what a species is. And so by and large, these hybridization events are rare. So like in the case of the Warbler hybrid, the Maggie and the Redstart that I talked about earlier, you probably expect that like 1 in 10,000 times, so really rare occurrences. And so it's not a call to alarm that things are shifting really fast or species are changing really rapidly right now. But it just makes the story more complicated. So scientists can't even agree on what a species definition is. And so hybrids make it even more complicated. And so that's why we want birders to be vigilant and keep doing their thing and report interesting hybrids that they think they see, because we are noticing patterns kind of in certain zones. Like there's a hybrid zone where Carolina and black-capped chickadees are known to hybridize. That doesn't necessarily mean that they're going to be lumped into the same species or that a new species is developing right before our eyes. By and large, hybridization are more rare occurrences. But yeah, it kind of shows the complexity of even the species concept itself.
Well, thank you for that. Here we go, Courtney. Paula says, well, she thanks you for the presentation and all that you do for the birds.
Oh, you're welcome, Paula.
until we see another question regarding hybridization, I'd like to go back to bird strikes, Courtney, because that's such a hot topic.
Yeah.
Not only in Cleveland, but in Columbus and Chicago. And as a matter of fact, at the Ottawa National Wildlife Refuge, we are embarking on a bird strike project. We're doing this because the National Fish and Wildlife Service is offering a grant to those, well, to refuges who do this, who conduct bird-friendly challenge projects to make the refuges friendly for birders. So we are going to be decorating the windows on the visitors center with all manner of artwork and some film, one-way film. And I'm going to be doing some bird strike monitoring. I'm going to kind of pioneer it. And what I'm interested in knowing is I know there are several places out in Northwest Ohio that will take carcasses and injured birds. But I talked to Tim and he said, you can just bring them to me on your way home, which is a 2-hour drive. But he said they'll survive, and if you properly handle them the way that he trained me to do that. So we are trying to make our stamp on the problem at the refuge where there is a problem. It's not a huge building and it's not in the middle of a city, but it's right on the migration path. And that's what makes it such a sensitive area for birds to do that. And another attendee says, this has been one of the best Zooms I've seen. Thank you. It has been one of the more scientific ones that we've had during this virtual festival that the Friends of Ottawa have sponsored. And I think there are many more to come. We don't have to belabor the audience because everybody has pretty much left. So when the last one leaves, Courtney, I'll ask you and Rebecca to hang on and we'll talk about how it went. There's somebody who's saying, yes, Judy is saying, what exactly should we do if we see a hybrid?
So I would eBird it. So if you don't have an eBird account or if you're not familiar with it, it is a free app. And I kind of got addicted to eBirding. Every time I go eBirding, it's kind of, you can log what you see. And so you can report an interesting bird sighting on eBird. If you are a Facebook user, there are plenty of birding Facebook groups. So if you think you see something really cool and interesting, try your hardest to snap a picture of it and start sharing it with your birding community and get the conversations going. And I've slowly but surely gotten better at taking a picture through my binoculars. With a little bit of practice, it can be done.
And Cindy says, thanks, excellent science delivered with good humor. So there's a shout out for you. We still got 22 people in the meeting room and listening audience. If you want to speak to Courtney, just press your space bar or unmute yourself and ask away. Just say hello or something.
I have a couple of questions, if you don't mind. My name's Christy Miskman. I actually work at Case Western Reserve University, and I am a scientist, and we do human genetics research. And I was just curious, when you're doing this, like, is there any volunteer activities or opportunities in the lab, like at the museum or anything like that? I mean, I have, transferable skills, and this has become sort of like a pet project, pet passion of mine. So I'd love to help,
Without like jumping out of my screen, I'm going to calmly say yes. It would be awesome to have you help out. We've got a couple projects where we just need to multiply the amount of hands and multiply the amount of hours in the day to get to. So yeah, I'll put my email in the chat. And if anyone has a question or wants to get involved or anything, feel free to email me. Let me make sure this goes to everybody. Yeah, go ahead and shoot me
There
an
it
email.
is.
I'd love to connect.
And one other question, as far as specimen are concerned, obviously you're curating all these. So when one of us happens upon a dead bird that we see, that like the other day I came on Case's campus, I think it was a strike, if I'm not mistaken. It was a little brown creeper, and the bird was in perfect condition. And I thought, oh my gosh, should I pick this up and try to give it to someone? So what do you recommend in something like that?
So that's a great question. So technically it's illegal to have a bird or any bird parts in your possession because they're all protected by the Migratory Bird Act, but it's kind of a weird gray legal area if you're going to bring it to a place like the Cleveland Museum of Natural History where we do have the permits to have those birds. And so if you find a dead bird and it passes an initial sniff test, if it doesn't pass a sniff test, it's too far gone and I don't want it. But if you find, like you described, like a perfectly beautiful looking bird that just is dead, if you write down the location you found it and the date that you found it, you can bring it to the museum. And we have so many folks that bring birds to the museum that we had to actually buy our security desk, our front security desk, their own mini freezer to take in the birds. And so, yeah, and actually on Case's campus, one of my volunteers, Owen Lockhart, works at Case, and he brings birds from Case campus to the museum quite often. And so, yeah, if you find a bird, try to get it into a freezer. Just put it in a Ziploc bag in a freezer until you can run it down to the museum. And just, yeah, those two pieces of information makes it a voucher specimen. We need to know where it came from and when it came from, and I'd be happy to take it. And then write your name down too, because then you're the official collector and then your name is associated with that bird forever, which I think that's pretty cool. But
So do I. Excellent. Thank you.
you're welcome.
All right, folks. Well, there's still a bunch of people in the meeting room. If you'd like to continue to talk, you're welcome to ask Courtney a question. Otherwise, Courtney, while people are still here and while we've got some time before at the top of the hour, regarding the taking of bird strikes to the museum, do they all have to go through Tim at Nature and Lake Erie Nature and Science Center?
So not necessarily. So the birds that go through Lake Erie Nature and Science Center are like officially a part of the Lights Out Cleveland data set, which we handle those birds a little bit differently from, say, a bird that just somebody finds and brings in. They all end up in the same place, but because of the amount of data associated with the Lights Out Cleveland birds, we can do a lot more analysis on these birds, and we actually treat it like its own data set. And so the Lights Out Cleveland birds go through Lake Erie Nature and Science Center and they get assigned a specific number. So it's a little bit different, but yeah, if you just find a bird and it's dead, we don't take injured birds. You want to take that one to the rehab. But if you find a dead bird, you can bring it straight to the museum and it'll end up in our research collection eventually. Okay,
thank you. Fred says, FYI, my wife's name is misspelled on a specimen at CMNH. Who should I speak to?
I think I fixed that problem, but if I didn't, email me again and I'll make sure that that's fixed, because I know,
okay.
I think I've dealt with that.
Real good. Another
double check.
Another
Okay.
viewer says, go ahead, Fred.
I just said thanks. I wasn't sure. I know I called it in, but we saw it on a tag a little while later. It was still misspelled. So
okay, I will double check next time I'm in there.
It's a red-throated loon.
Oh, okay. We don't have that many of those. They'll be easy to find.
Okay, thank you.
Courtney, there's a question here. Are lighthouses much of a problem for bird strikes?
Not so much as buildings. So it's artificial light from buildings and from street lights that are the huge problem. Lighthouses, they're kind of few and far between where they do exist. And you, Tom, you mentioned that you're doing the project with the Lights Out, or a window strike project on a property that's kind of not in a city. And although downtown cities are very dangerous, residential buildings and houses are actually the biggest threat to birds. So, so yeah, if you notice you've got a window at your house that's an issue with bird strikes, you should deal with that window. It'll definitely help. But I don't think lighthouses are a huge concern. It's more the lights at night, the reflective glass, and house cats aren't doing birds any good as well. And then I saw Judy's got a question. I live in the Akron area. Are there a place in Akron that collects dead birds for research? So actually there is a Lights Out Akron group. And so I would suggest you look into that group and see how you can get involved or hand the birds over to that group. And so that's a encouraging thing is Lights Out groups are popping up all over the country, and it's very encouraging. So I would suggest you reach out to those folks.
We still have some people in the room, which indicates there's still an interest, and not seeing a new question. I wanted to mention that, or a comment on something you just said, Courtney, about if you have bird strikes in your home, to do something. And that's the key issue. The elephant in the room is, what do you do? And what we are doing at the refuge is using different kinds of materials to obscure the windows, like bird tape and dots.
Yep.
Mixes of dots and tapes. Also, Zen curtains. We're also using tempera paint, and we're using children to do their own artwork on the lower windows. Zen curtains, screens for the insides of doors, and then of course the one-way window film with murals or bird designs. So all these things are strategies you can use to make your own home more bird friendly. They're low cost. You just go on the website and find them. Or if you're really, really interested, you can contact Courtney or me and learn more about that.
Yeah, and it's encouraging that this has become more and more of a talked about phenomena. And so the resources to help are just becoming more and more available and cheaper. The quickest and cheapest and easiest thing that I suggest is just take a bar of soap and scuff up the window. Just do anything to break up that reflection. Because I have a problem with cardinals trying to defend their territory against their reflection in the window. And so you just take a little bit of soap, and then I feel like I clean my window a little bit when it rains. And it gets the cardinal to kind of understand, like, okay, go waste your testosterone some other place and maybe not at your reflection all day. So yeah, it's all about just breaking up that reflection. And there's very expensive options and there's very cheap options, but just kind of putting forth the effort would do a world of good. And yeah, birds only have a couple things on their mind in the spring. And if you see a cardinal, especially cardinals for some reason around my neighborhood, if you see them going to town on their reflection, maybe help a fella out and scuff up that window with some soap or some stickers or film or stringing lights or just string rather. There's lots of simple ways to get the birds to go be a bird somewhere else.
Courtney, I want to add a rule of thumb that we're using, and that is called the 2x4 rule. So if you're going to hang something or draw something, even put bar soap on the window, you want to make your lines 2 inches to 4 inches apart, vertically or horizontally. And the reason it's 2 and 4 is the 4 indicates the width or the wingspan of a bird, and the 2 inches is generally the height, the dimension of a bird flying straight into a window. If the lines are 2 inches together, they may be distracted from going into the window because they won't think that it's they can get through it. So the 2x4 principle is important.
Yeah, that's a great point. Thank you.
You're welcome. Well, okay, 13 people left, and it looks like we're kind of out of steam, folks. So I'm going to suggest that we close the meeting.